In today's notes, we're going to connect some ideas from our last unit of cells with our new unit genetics. So just as a reminder, eukaryotic cells have double-stranded DNA. So they have DNA, it's double-stranded. I'll talk more about what that looks like. It's linear, meaning it's like one long piece of DNA, so it's like a one long piece of string. And it's found in an organelle known as the nucleus. So the nucleus has its own double membrane that goes around and it houses that DNA. The nucleolus is also inside the nucleus. That's the site of rRNA production for ribosomes. The nuclear membrane has nuclear pores that let very small molecules travel in and out of the nucleus. The rough ER helps move proteins and other molecules around the cell. Ribosomes are the site of protein production. Remember, there are free ribosomes that float, as well as ribosomes connected to the rough ER. The smooth ER helps with lipid production. The Golgi apparatus marks and transport proteins and other molecules that go around or out of the cell. And then we have the mitochondria, which were really interesting if you remember, because they make their own ATP, so they make ATP for the cell. They also have their own circular DNA. That DS stands for double-stranded, so they have their own circular double-stranded DNA. They have their own ribosomes, and they can divide and replicate on their own, which are interesting attributes for an organelle. Prokaryotic cells, if you remember, are cells like bacteria. They have DNA that is also double-stranded, but in this case it's circular, so they connect on their two ends. Unlike linear DNA. So circular DNA would be more like a rubber band. This DNA is found in the cytoplasm, so there is no organelle known as the nucleus here. And we also potentially have plasmids, which are just known as accessory pieces of DNA, and they tend to be very, very small. So this is sort of a very loose drawing of our prokaryotic cell. And just remember, prokaryotic cells are much, much smaller in comparison to eukaryotic cells. So we talked a little bit about both of those cells having DNA. Now let's talk a little bit about what DNA is. So if you remember back to our biological molecules unit, we talked a little bit about the building blocks of nucleic acids. So DNA is a type of nucleic acid. If you looked up here, it stands for deoxyribonucleic acid. So it's a type of nucleic acid. DNA is double-stranded, so I'll show you what that means in a little bit. Sometimes referred to as a double helix or a twisted ladder. They have what are known as anti-parallel strands, and the building blocks are known as nucleotides. So let's talk a little bit about what the nucleotides are. These are the monomers or the building blocks of our DNA. Our nucleotides consist of three parts: the phosphate group, the sugar, and the nitrogenous base. So the sugar is called deoxyribose. I know it's a sugar because it ends in that OSE. And it typically looks like this. This would be the sugar for RNA. We will talk about that later. There is a phosphate group. That just means a group that has a phosphorus in the middle. And there are four nitrogenous bases. So adenine, guanine, thymine, and cytosine are our four nitrogenous bases. This is sort of a loose structure of what those guys look like. You do not have to have that memorized by any means. But you do have to know the bonding rules. So adenine typically likes to pair up or bond with thymine, and guanine likes to pair up with cytosine. So what does that look like when we look at our DNA molecule? So what ends up happening is we've got one strand, and that's on this side. So these are all bonded together covalently. And then we've got a second strand on this side, and these are all bonded together covalently. And those bonds back here make up what's known as the sugar-phosphate backbone. So phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Same thing on this side. Those covalent bonds make up the sugar-phosphate backbone on this side. So it goes sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And in the middle, these ladder rungs, the ladder part of our DNA, are where our nitrogenous bases make these sort of loose hydrogen bonds with each other, and these are where our pairing rules come into play. So adenine likes to pair with thymine, cytosine likes to pair with guanine, and so on and so forth. So you'll see that my building blocks are all present. So here is one nucleotide, it's bonded to another nucleotide, bonded to another nucleotide, and then here's the second strand on this side, and these are all bonded together. Oh, I circled two. And so I've got two strands of these, and then they make these hydrogen bonds in the middle, and that's what connects my two strands. So DNA is always considered double-stranded. And so that's a little bit about what our DNA looks like. Normally you'll see it drawn like this picture over here on the left. Typically because it's a much easier picture to draw, but that's what those little ladder rungs in the middle are representing, are these nitrogenous bases, hydrogen bonding together. And then these rails on the side, that's representing those sugar phosphate backbones. So just as a sort of vocabulary point, when we're talking about how long our DNA strand is, we refer to it by how many base pairs long it is. So a base pair is two bases, nitrogenous bases paired together. So if I look at this particular segment of DNA, it is 1, 2, 3, 4, 5, 6 base pairs long. I've drawn a picture here that is 6 base pairs long. This picture over here might be, I don't know, like 30 base pairs long. So I would count how many rungs to my ladder there are, and that's how many base pairs I'm counting. And that's normally how we talk about our length of DNA. All right, so let's keep going. A lot of that should be review. Some of it is going to be new. But what I want to talk about is a little bit about how the DNA starts to organize itself into those chromosomes. So we talked a little bit about chromosomes when we talked about mitosis and meiosis. So a chromosome typically looks something like this. So remember, a chromosome is usually made up of one chromatid. If you look at a really good picture of a chromosome, what you'll see is these little bands, and it's been stained so that certain genes sort of pop up and we can see the color in them. But this is what a typical chromosome looks like. And so when the chromatid replicates, you get that X shape. So this is a chromosome with two chromatids. And what a chromosome really is, it's DNA that's been wound up really tight around these proteins. You don't have to know the name of these guys. All you have to know is that chromosomes are just super, super tightly wound DNA. So it winds up around these proteins and it coils up and it makes a really, really tight shape and it forms these chromatids. So it's almost like how they twist yarn up. So they twist it and they roll it and they coil it and they make it into a nice, tight sort of like ball bar thing. That's basically what a chromatid is. It's all of that long DNA that can be millions of base pairs long, all wound up super tight into a chromatid / into a chromosome. So when it starts to unravel, right, it gets loose, we see that our DNA is going to be very, very long. Now, another new term might be the term gene. So a gene is a segment of our DNA that codes for a particular protein. So on our very, very, very long DNA strand, we might have segments of it that code for actual proteins. And those proteins will do their jobs, and then we'll see our observable traits. And I'll give you guys some examples of those later. And then in between the genes, there might be non-coding regions of DNA. But our DNA is very, very, very long. It's coiled up super tight into chromosomes. And not only is our DNA really, really long, but if you take particular segments of it, they might code for proteins, and those segments of DNA are called genes. So that's a little bit about what a gene is. So I've drawn another picture down here, and I've defined it specifically, segment of DNA that codes for a particular protein. And again, here's a very long DNA strand, and here is maybe a segment of it that codes for, we'll say, gene 1. Then there might be a non-coding region, and then we've got another segment that codes for another protein. The reality is that genes are actually a lot longer than this. They can be hundreds to thousands of base pairs long. I think the average human gene is something like 10,000 base pairs long, so they're very, very long. And that some genes have more than one variation, and these variations are known as alleles. So different versions of the same gene are known as alleles. So an example would be blood type. So there are different types of blood type that are coded for by different alleles. So one of the genes or one of the alleles that you could have gotten is for type A blood. Or maybe one of the alleles you inherited was for type B blood, or maybe one of the alleles you've inherited is for O blood. And you get to receive 2 alleles, one from each parent, but these are the different alleles that you could possibly get. So what I'd like you to do is, based off of what I've just covered and what you previously knew, in your own words, try to describe the relationship between a cell, the nucleus, DNA, chromosome, gene, nucleotide, and allele. It does not all have to be in one sentence. I'm going to guess that it's going to take 3 or 4 sentences in order to describe the relationship between all of these.